Assalamu alaikum. Welcome to the platform of Sindh Biotechnologists Association. Here I am your presenter Muhammad Fida Rehman. Today the topic of my discussion is on ELISA. Here are the main points of ELISA which we have to discuss here. Its introduction, principle, steps, types and applications of ELISA. What is ELISA? ELISA is enzyme-linked immunosorbent assay. It is a sensitive plate-based assay. The plate used in this technique is made up of polystyrene having 96 wells. This technique was first introduced in 1971. What is the major use of this technique? This technique is used to detect and quantify substances such as antigens, antibodies, proteins, glycoproteins in different biological samples. Next, what is the principle of ELISA? In the ELISA, an antigen or antibody is immobilized to the solid surface. This solid surface is of the microtiter plate wells, where it forms antigens and antibody complex. The detection of this antigen or antibody complex is determined by the activity of enzyme conjugate via incubation with substrate to produce a measurable product. After adding the enzyme conjugate, this enzyme conjugate attached to the surface of the antibody or antigens, then substrate is added. This substrate react with the enzyme conjugate and produce products. Here are the main steps of ELISA. Antigen or antibody coating, next one is blocking, incubation, and last one is developing solution. In the first step, antigen or antibody coating, the antigen or antibody is coated to the surface of the wells. These antigens or antibody is made up of protein, peptides or crude proteins. Their choice depends on the nature, which kind of antigen or antibody we needed. These antigens or antibody attached to the surface of the microtiter plate is due to the hydrophobic interaction between protein and plastic. The next one is blocking. Blocking is used to prevent the surface of the wells from the non-specific binding. For this, the blocking buffers are used. These blocking buffers are PBS, phosphate buffered saline, TBS, tris buffered saline. Blocking solutions are made up of proteins which attach to the surface of the wells to prevent the surface of the wells from the non-specific binding after antigen or antibody complex formation. The next one is incubation. In this step, the antigen or antibody complex which form already is incubated with a primary and secondary antibody having enzyme conjugate. This enzyme conjugate is HRP, horseradish peroxidase or alkaline phosphate. Mostly these two enzymes are used in the process of ELISA. The next is developing solution. In this step, the substrate or chromogen is added. This substrate or chromogen is attached to the enzyme conjugate and produce colors. The substrate or chromogen is mostly TMB, tetramethyl benzidine. Now next, what are the types of ELISA? There are four major types of ELISA: direct, indirect, sandwich and competitive ELISA. Let's talk about each type one by one. The first one is direct ELISA. In the direct ELISA, the antigen is coated to the surface of the microtiter plate wells with the process of passive adsorption. After the attachment of antigen to the surface of the plate, then a primary antibody which is coated with the enzyme is added. This primary antibody attached to the surface of the antigen. Then after incubation, a substrate is added which react with the enzyme conjugate, then produce signals. These signals gives color for the positive results and color doesn't appear for the negative result. Next is indirect ELISA. In the indirect ELISA, the antigen is already attached to the surface of the solid plate. Then a primary antibody is added. This primary antibody attached to the surface of the antigen. Then after incubation, a secondary antibody which is conjugated with the enzyme is added. This secondary antibody attached to the surface of the primary antibody. Then after again incubation, a chromosome or substrate is added which gives a signal for the positive results and the signal doesn't appear for the negative result, and they are read in the reader at 450 nanometer wavelengths. Now next is sandwich ELISA. In the sandwich ELISA, the wells are already coated with the capture antibody. Then antigen is added which attached to the surface of the capture antibody. Then a secondary antibody which is called detection antibody is added. This detection antibody attached to the surface of the antigens, and now antigen is sandwiched between two antibodies. Then a substrate or a chromosome is added. This substrate reacts with the enzyme conjugate and gives signal for the positive results, and the signal doesn't appear for the negative results. Now next is competitive ELISA. In the competitive ELISA, there is a sample tube which is already have antigens and antibodies. Then a label antibody. This label antibody is called due to the enzyme conjugate which attached to the surface of this antibody is added. This enzyme conjugated antibody attached to the different antigens of the samples and form complex. Then these antigens and antibody complex are removed through the process of washing. Then only the antigens and antibodies are left which are not attached to the antigens of sample. There are reference antigen which attach to the surface of the wells. Now only the antigens are only the antibodies are left which doesn't attach to the surface of the antigens of sample tube. These antibodies attach to the reference antigen and form complex. If then a substrate is added. If the signal is weak, it means that signal weak mean color is low. Color intensity is low. If the color intensity is low, it means that the reference antigen and antibody form less complex as compared to the complex form in the antigens of sample and the antibodies. And if the color intensity is more, it means that the reference antigen is more in number as compared to the sample antigens. There are many applications of ELISA, but here we shall discuss only its application for the diagnosis of hepatitis. We will detect hepatitis E and hepatitis A through the process of ELISA. In the detection of hepatitis E, the technique of ELISA used is an indirect type, and the antibodies which we have to detect is of IgG antibody. The microdata players are coated with the HEV recombinant antigens which are specific to the IgG antibodies. The serum or a plasma in the sample tube is first diluted in a diluent, after which only a specific amount of this diluted sample is added into the wells. If the antibodies are present, then they are captured by the antigens. After incubation, the plates are washed to remove all the components except the bounded antibodies. Only those antibodies, IgG antibodies complex, are remained. Then antigen antibody complex is detected by the anti-HIgG antibody, which is called secondary antibody, which is conjugated with the enzyme, and the name of this enzyme is a horseradish peroxidase, which is added. After second incubation and the washing, then a chromosome is added which react with the enzyme conjugate to convert it into a colored product. The color intensity is directly proportional to the amount of the IgG antibodies in the sample. If the color is more, it means that the IgG antibodies are more in number. And if the color intensity is less, it means that the IgG antibodies are less in the sample. Now next we shall discuss about hepatitis A. Here the antibody used is IgM, and the technique used for the detection of hepatitis is a sandwich ELISA. The microplates are coated with the antibodies which are directed to the IgM proteins. The serum or plasma is first diluted in a diluent, after which only a specific amount of it is piped into the wells. If anti-HAV IgM antibodies are present in the sample, then they are captured by the peri-coated antibodies. These antibodies are coated on the surface of the wells. Then incubation takes place. After first incubation, the plates are washed. To remove all the components, only the IgM antibodies are left which attach to the surface of the well. After second incubation and washing, a chromogen substrate is added upon which the enzyme reacts. It means that a chromogen is added which react with the enzyme conjugate and a color is produced. The color intensity is directly proportional to the amount of IgM HAV antibody in the sample. It means that if the color intensity is high, then the IgM HAV antibodies are more in numbers in the sample. And if the color intensity is low, it means that the IgM HAV antibodies are less in number in the. Thank you so much.